I want to turn our attention now to the contractile cardiomyocytes, and these are striated muscle tissue. You probably already knew that. Striated meaning that you have the I bands, the H bands, the A bands, etc. So the structure of the cardiomyocyte that is contractile internally is somewhat similar to what you saw in striated or skeletal muscle, so that you have your actin, you have your myosin, your S1 head, and the ratcheting action with the binding of calcium and such. So all of that is very similar, so I'm not going to rehash that part of it. So if you need a refresher, I'll just encourage you to go back and review your skeletal muscle lectures and notes. But there are a number of distinctions between the cardiac muscle, contractile muscle, and the skeletal muscle. The skeletal muscle, as you'll recall, is not made of discrete cells, but is made of a muscle fiber. So recall that you have the nuclei are on the periphery, and the fiber is a functional unit. Well, when it comes to cardiomyocytes, they are discrete cells. So what you have here, and you can see the nuclei are in the center, and you also see from the cartoon that the cardiac muscle are not little simple rectangles. They have a branchy pattern to them. The reason they branch is that enables for one cardiomyocyte to be in contact with a greater number of neighboring other cardiomyocytes. So the branching pattern is there for a reason. Another distinction that you see in the cardiomyocytes that are contractile is that you have what is called an intercalated disc. And so you see the little intercalated discs here, and here's just a zoom one. So there's your intercalated disc there, and it's just a way for one cardiac muscle cell to interface with the other. So let's take another look at the intercalated disc and see what is making them up. So I've already kind of drawn out two cardiomyocytes, cell 1 and cell 2. And so I'm going to draw several structures here. So one structure, I'm just going to go ahead and just draw here and here. And so we have these structures here, and then we have some intermediate filaments. There. So I'm going to say intermediate filaments, and they're very strong cytoskeletal structures. And then we have some specialized proteins that are going to be connecting through here. So this first structure that I've drawn, we're calling a desmosome. And if you've had histology, then you know what that is. A desmosome is a structure that allows two cells to be strongly bonded to each other. So these two cells here, by virtue of the proteins in the middle, are connected to the cytoskeletal structure of each cell, and in particular the intermediate filament. And so this is what that is. The proteins in the middle here are called cadhedrons. Okay, so that's what those are. The next thing I want to draw is, I'm just going to draw it over here and here. Okay, and so this one, again, I have cadhedrons in the middle here, and then I'll just go maybe a different color, okay, here. And what I have here are actually microfilaments. So I'm going to go microfilaments. In particular, these are actin. So actin actually interfaces here, and so this is the actin that's going to be the beginning of the actin-myosin interface, so that when you have the ratchet action, it's actually going to draw the cells to be smaller. But again, you still have the cohedrons in the middle to kind of keep it together. Another structure that you're going to see in the intercalated is going to be here. And these are going to be gap junctions. And these gap junctions are made of these specialized channels, okay, and the protein is called connexin. And they're made of 6 units. And what their characteristic is that they're voltage sensitive, and so given the appropriate voltage potential, they can open and close. When they're open, of course, creating a channel, which then allows ions and small molecules to actually go from one cell to another cell. So this is a mechanism by which one cell can exchange material into another cell, and what we're going to find out in a moment, that typically the main players here is going to be calcium and sodium. So they can go through that. I will also mention that there are many, many, many isoforms of connexin throughout your body, because the cardiac cells are not the only cells in your body that have gap junctions. You have them in smooth muscle, etc. So one connexin is not the same as another connexin, so forth, so on. Some will open fast, some will open slow, some will stay open longer, some will stay open shorter. So again, all kinds of variations functionally of the connexins. But the connexins basically make up the gap junction. So those are the main players here, and I'm left this other one open because I'm going to come back to it later. But this little area here, the widened area right next to the gap junction, is called the perinexus. And of course, the nexus is going to be the gap junction area. So this is a very important area. I'm going to talk about that in a moment. Okay. So the reason you have this is to, again, the intercalated disc is to give some integrity for the cardiomyocytes such that when they contract, they don't pull apart. Because what I just told you is that you have ions that are going to be flowing from one cell to another cell, and if you don't have these types of structures here that help the cells to adhere, then when one cell contracts, pulling from the other cell, it literally will tear the gap junctions apart. So the function of the intercalated disc is to strengthen the contact so that the cardiac cells don't literally tear each other apart and, of course, destroy themselves functionally. Now, let me make another point here. You'll recall when we were talking about neurons, okay, so neuron, axon, and then another neuron, axon, so forth, so on, that when you have an action potential, okay, and it is propagating, what you have here is a series of voltage-sensitive sodium channels that open, and then one trips the other one off and trips the other one off, kind of like falling dominoes. I've used that analogy before in terms of each group of voltage-sensitive sodium channels bringing the next group to its threshold, and then it opens up, so forth. And again, remind you that the voltage-sensitive sodium channels are double-gated, and a point that I'm going to make again later. There are many, many, many kinds of voltage-sensitive sodium channels. In fact, in cardiac tissue, there are at least a half dozen isoforms or different varieties of voltage-sensitive sodium channels. They all have their own characteristic. But then again, everything we talk about, there's different varieties of them. So just want to remind you of that. So you'll recall the challenge with neurons is you can have an active potential going from voltage-sensitive sodium to voltage-sensitive sodium, but when you want to go to the next cell, of course, action potential dies. So you know that you have a chemical synapse, releases neurotransmitters, and then communicate with the next cell down the line. So the connection here, again, a chemical synapse. You don't have the same challenge when it comes to a skeletal muscle fiber because it's all one fiber. So you have these voltage-sensitive sodium channels throughout the fiber. So once the potential gets going in the muscle fiber, the entire fiber can contract because it is a structural syncytium. So when you think about the cardiomyocytes, okay, and I just got through telling you they're not square or rectangle, but I'm going to draw them anyway just to make things simple. All right, you got that. So I have then voltage-sensitive sodium channels, right? So if I have an action potential moving in that direction there, when it gets to the end of the cell, the action potential is going to die. So you don't really have a way of, a voltage-sensitive channel tripping off this voltage-sensitive channel here. At least that was the theory. So you have to have another way of communicating the fact that one cardiomyocyte has depolarized, and how is it going to connect with the other. This is where the gap junctions come in. And so what you have here, these little gap junctions are so typically associated with the intercalated disc. Now, they don't all have to be associated with the intercalated disc, but a lot of them are. So intercalated disc, and so what's going to happen is when you have an action potential here, it's going to open up those connections, those gap junctions, and then typically you're going to have calcium and sodium flowing in, and the movement of the positive charge into the next cell is enough to bring then the voltage-sensitive sodium channels in that next cell to threshold, and then you have an action potential there, and so forth, so on. So you're actually hopping action potentials from cell to cell, but you see the critical role that the gap junctions play to be able to communicate from cell to cell. Another thing that I want to point out is because you have so many different varieties of connections, the type of connection is characteristic can have a significant influence on the conduction velocity. So when I say conduction velocity, typically I'm going to be writing CV means conduction velocity. And we're going to find out in future lectures how important having the appropriate conduction velocity is as you have a depolarization of your cardiac tissue. Okay, so there's that. And on the next page, again, another cartoon, a prettier picture of the connection and the gap junctions. So there's my gap junction there. I'm going to say G or GJ, gap junction here. And here is the perinexus. Again, it is that little space that is right next to the gap junctions. Now, both contractile and non-contractile cardiomyocytes communicate via gap junctions. But the intercalated discs are only found in the contractile part of the tissue. So you don't expect to find intercalated disc associated with the AV node, SA node, bundle of His, and the fibers that are running down the septum, et cetera, or the Purkinje fibers. So the intercalated disc appear when you have the contractile part of the heart muscle. Okay, so for many, many, many years, that was the convention for how one cardiomyocyte communicated with another cardiomyocyte. But there was always anomalies in the data that suggested that the story was more complicated than that. And it turns out it is. So I want to introduce you to another concept. And it is the concept of ephaptic conduction. Ephaptic conduction is yet another way for one cell to communicate with another cell when we're talking about voltage changes. So the way this works is, and I'll try to cartoon it out for you. Suppose we have a cell here, cell 1, and then over here we have cell 2. And suppose you have some type of depolarizing event in cell 1. So that means that the membrane potential, of course, has changed. And so the depolarization in cell 1 creates a change in the electrical field around it. And if you have cell 2 that's very close to cell 1, you actually could have the situation where the change in the electrical field can alter proteins in the adjacent cell. So more specifically, you have a depolarizing event in cell 1, and you have voltage-sensitive sodium channels in cell 2, and the fact of depolarizing in cell 1 changing an electric field is enough to bring the voltage-sensitive channels in cell 2 to threshold. That's a different scheme. So here is a prettier cartoon that really presents kind of as a puzzle. So what we have here is our traditional gap junction, and so these guys here are going to be my connexins. And of course, they're going to open and sodium and calcium will be flowing through. So that certainly is one way of communicating a charge between two cells. Then right next to it, during the perinexus or the perinexus area, what they've discovered is that you actually have voltage-sensitive sodium channels. They're just sitting there, a certain type of voltage-sensitive sodium channels, and they're connected with some other proteins across here, whose term is I'm not too worried about. And so this area here conceptually is called the cardiac EFAPs. And so let me walk it through. This was a curious type of experiment that they did, in the sense that when they experimented on this tissue and everything is normal, then the normal rhythms were occurring and everything was fine. But they were able to manipulate the cellular structure such that they now separated, made wider, the perinexus. And when they did that, they generated an arrhythmia. In other words, the heart did not work appropriately. The gap junctions were kept in place. Everything else was kept in place. All they did, through some clever manipulation, was widen the perinexus and things went sideways. So what that then suggested was there's something going on in the perinexus that doesn't directly involve gap junctions that's very important for normal functioning of a cardiomyocyte. And so what the other experiments were done, and what the evidence looks like now is that you in fact have ephaptic conduction, such that you have one cell here, cell 1, and of course over here is cell 2. So when you have a depolarization or an action potential of cell 1, this field here will actually open up, the change of the field will open up those voltage-sensitive sodium channels there, allowing sodium in that were in the perinexus space. And that in fact could also aid in bringing the adjacent cell to threshold. So what it looks like now is that you have a more complex system where you have an electric synapse, gap junction, working along with an ephaptic conduction process, which is really, really cool. So here is another cartoon. Now you can read the words down here, but it's kind of slick. So in A, what they're looking at is just the standard model for your gap junctions. So connexin and connexin 43 is a very popular one in the cardiomyocyte. There are other isoforms in the cardiomyocyte as well, but 43 seems to be one that's fairly common. So again, and this is cell 1, cell 2, so you have a depolarization, and then you have sodium coming in, and then that then in turn is going to cause the gap junctions to open, and when it opens, now you're going to have your sodium and your calcium flowing in that brings the next one to threshold. And so by the virtue of having sodium calcium flowing in, you can see there that you're now having a rise towards the threshold for the cell 2, and of course that then is going to trip open that, and then you have your sodium rushing in, and here goes the depolarization in cell 2. So that's been for many, many years, that's the standard model. So what they're finding out now, though, in a B type of scenario, and go down here. All right, so now what we have is we have another scenario here. So this right here is going to be the perinexus. So this that space right there is the perinexus. So now what you have, again, cell 1, depolarization. But with the sodium coming in here, now you're depolarizing that cell. That in turn is going to be altering the electric field. First of all, what you're going to have is a depolarization here, and then you're going to have sodium running in here. So the sodium is going to be opening up, so I'm going to have sodium leaving this perinexus area. So the mere fact that I'm having sodium leaving this perinexus area here, so I'm losing positive charge, and if I'm losing positive charge in this area here, that is going to in turn make this to be more positive. So this area here, because I'm having sodium channels or sodiums going through those voltage-sensitive sodium channels in cell 1, so this area here is becoming more negative. And if the perinexus is becoming more negative, then what that means then is that this area here is becoming more positive. And if it's becoming more positive, then it has the ability to bring that voltage-sensitive sodium channel to threshold, then allowing sodium to rush in, and in here we go, in terms of initiating an action potential in cell 2. So yet another way for the cells to communicate. Of course, this is not either or, so the latest thinking on this is that it is a combination of these two mechanisms. So we actually have a mixed mechanism. So we have ephaptic coupling and gap junctions that are going to be involved in the axial potential propagation from cardiomyocyte to cardiomyocyte. Now, just a reminder, and I'm going to elaborate on this a little bit later before we close this up, that when we were discussing non-contractile cardiomyocytes, such as the cells in the SA node, AV node, bundle of His, or whatever else, the voltage-sensitive channels were not players. So your undulating axial potentials came exclusively by way of calcium influx. So that is true through AV node, SA node, bundle of His, etc. So that is in contrast to a contractile cardiomyocyte, which is, we're going to see this very clearly in the next lecture, where the initial event depolarizing that contractile cardiomyocyte is going to be voltage-sensitive sodium channels, which will in turn open then L-type calcium channels. So the big trigger in the contractile sodium voltage-sensitive channels, where you don't have those in the non-contractile. Now, there is one exception to that. There's always an exception. One exception are the Purkinje fibers. Okay, the Purkinjes actually do have voltage-sensitive sodium channels that aid in their depolarization. Okay, so this is a kind of cool stuff in terms of it was a little more complicated than what for years they thought it would be, but this is, anyway, it's just neat, and so just wanted to make sure you understood how complicated these systems can be, and also how things change. Because I could not have given you this lecture 20 years ago, because they had no idea about that other mechanism. But in the past 10 or so years, there's been tons of data suggesting that this, in fact, is a very complex system in terms of how you communicate a voltage potential change from one cardiomyocyte to another cardiomyocyte. Okay, so we'll pick up next time.